All right, so our next section here in this unit is cellular respiration. And so we talked about this back in 7th grade when we studied both these things. And we went over just really the basics of what cellular respiration is, the basic formula for the overall reaction. Obviously, in this section, we'll get into much more of the detail about how cellular respiration actually works, where these various things happen, what are the chemical reactions that take place that allow it to happen. But just if you were to give a general statement, what is respiration? What would you say? So what is respiration? So yeah?
It's actually making the sugars here, like glucose, out of like ATP and stuff like that.
You sort of got it reversed there. So it's using glucose to what? To produce ATP, which is the currency of energy that cells actually run on. Okay, and that's really what respiration is. It's taking that glucose and extracting the energy from it, because we know it has energy stored. Where is the energy in a molecule stored?
Mitochondria.
Well, it's released from the mitochondria, but in a molecule.
In the bonds.
It's in the bonds between atoms. Okay, those bonds, when we break those bonds, some of that energy can be harvested. So you can think about cellular respiration as transferring energy from chemical bonds in glucose into chemical bonds in another molecule, ATP. And those bonds storing energy of ATP are what the cells actually use. Hello? Hi.
Who's a chicken?
They're not it. This is
Why did the
something
turkey
else.
cross the road?
They weren't here last year.
Because he wasn't a chicken.
Okay? Yeah. So,
I don't understand why the chicken
glucose, a sugar, has energy in it. In fact, sometime I'll show you a video, but you can release that energy by burning sugar, and it gets released as heat and light. But heat and light are not really that useful for living organisms. So instead of a sudden release of energy from glucose, respiration takes that energy and sort of takes it a little bit at a time and turns it into types of molecules that cells can actually use. So it's a whole series of reactions that are controlled by a whole bunch of different enzymes, sort of gradually releasing this energy. And there are really two main types of respiration. Aerobic respiration and anaerobic. You know what those terms mean? What does aerobic mean? Sebastian? Uses oxygen. Aerobic uses oxygen. Anaerobic is respiration that happens without using of oxygen. So we see here our molecules. We have glucose. We know 6-carbon sugar. Carbons arranged in a ring. And those bonds between those atoms store the energy. And here's another molecule, ATP, which again has bonds between molecules. What does ATP look like? Does it look like another molecule we've talked about? Cormac?
The nucleic acids.
Yeah, it looks like a nucleotide, right? Remember we had, what is adenine? No? Adenine's one of the nitrogenous bases. What is ribose? A sugar. Okay. What else do we have here? Phosphate. So we have phosphate, sugar, nitrogenous base. Basically, we have a nucleic acid. What type of nucleic acid? Is it a? Yeah, it's one of the building blocks of RNA. Except to form ATP, we add on some other phosphate groups. And as we add on these other phosphate groups, these bonds between the phosphates is where the energy is stored. And these bonds are broken sometimes and then replaced at other times. So let's talk first about anaerobic respiration. We're not going to get into many details here about anaerobic respiration. Most of our material is about aerobic respiration. But anaerobic is respiration without the use of oxygen. Okay? And it takes place in several different types of living things in various conditions. So some organisms lack the enzymes. So there are times when there's just no oxygen available, and an organism would go through this process of anaerobic respiration. Other types of living things just don't have the enzymes. They're not equipped for aerobic respiration. And so anaerobic respiration occurs in the cytoplasm of a cell. And it starts off just like aerobic respiration with a process that's called glycolysis. Okay? And glycolysis, like we said, takes place in the cytoplasm. And glycolysis takes a molecule of glucose. Some energy is added here. It takes actually 2 ATP. Some energy has to be invested. And then that glucose molecule is oxidized. It's broken down, and some of the energy is harvested. It's broken down into 2-pGal, which we recognize from photosynthesis. It's further broken down into 2 molecules of what we call pyruvic acid. And the end result is the production of 4 ATP molecules. But why did I say here that it's a gain of 2 ATP molecules? Caleb?
Because it starts with 2.
Yeah, because it takes 2, you have to put in 2, and then you get 4 back out. And so the net gain is you gained 2 ATP molecules. So you gained some energy. Okay? And that's the process. Nate?
In the workbook about like 2 kinds of like gain, like net gain and something else. I forgot what it was. Yeah, overall.
I'll have to look at it. I don't remember the diagram they referred to, but we're going to go over that, so we'll talk about it. Okay, so that's what anaerobic respiration, part of it, is the oxidation of glucose to form those pyruvic acids, and then they can further be broken down. Okay, and we call this process fermentation. So this pyruvic acid that's produced in glycolysis, it still has a lot of energy left in it. Going from glucose to pyruvic acid, it gets some of the energy out. You got 2 ATP molecules, but there's still a lot of energy left in that molecule. So it needs to be further broken down to extract some more of that energy. And it happens through fermentation. And there's several types of fermentation. And some bacteria can take this pyruvic acid and further oxidize it and turn it into lactic acid, okay, and extract some more ATP out of it. In some animals, when they don't have enough oxygen, okay, then they can switch from aerobic respiration into anaerobic respiration and do these steps to get the ATP. And so some examples, bacteria. If you look, if you eat some yogurt this morning, if I look on the container of this Chobani, what I'll see here, it says contains live and active cultures of S. thermophilus, L. bulgaricus, L. acidophilus, bifidus, and L. casei. So it has several bacteria. That's what yogurt is, a fermented food product, like many cheeses. If you've ever had sauerkraut, kimchi, these are all foods that are produced through the use of fermentation. And what happens is the bacteria consume this pyruvic acid, and they get some of the energy out. Okay, and it forms lactic acid. In our muscles, this happens. So when you're sprinting, for example, you're running, and for a while your muscles are going to be going through this process of aerobic respiration for your muscles to get the energy they need. But at some point you run out of oxygen. Your breathing can't replenish the oxygen that your muscles are using at the rate that you're using it. So the oxygen level decreases and decreases until it's basically gone. Then the cells of your muscles switch. They switch from aerobic respiration, because now there's not enough oxygen, to anaerobic respiration. And part of that process produces lactic acid. And that's one of the reasons we get muscle fatigue. As that lactic acid in our muscles builds up, it burns, okay? Our muscles become fatigued, and eventually we have to stop that activity. You can't just keep going and going when you don't have enough oxygen. Once you stop that, exercise or whatever, your circulatory system helps remove that lactic acid that's built up in your muscles. You start to regain your oxygen balance, and then can switch back into aerobic respiration. So it happens in certain conditions in lots of living organisms. Another form of fermentation that results in different products is, an example is yeast. Yeast go through the process of anaerobic respiration and fermentation. They take that pyruvic acid that's produced, and then they convert it into other products. They convert it into ethanol, ethyl alcohol, and carbon dioxide. And again, this is something that we use in creating of foods and beverages. Beer, wine are fermented things where you add yeast to a mixture of whatever, grape juice or malted barley solution, and the yeast then digests those sugars present in those raw materials, and they produce carbon dioxide and they produce alcohol. Bread, when you add yeast to dough, you mix it around and let it rise, what's happening is the yeast are consuming sugars from that dough, from the flour, and as they release carbon dioxide, it causes your dough to raise, because there's lots of bubbles that are forming inside of that dough. And when you slice a slice of bread, those little air pockets you see in there, they were filled with carbon dioxide that was a product of yeast fermentation. So it's another example. So if we look here at sort of an overview, okay, if we talk about respiration, fermentation, they start off the same. Both start with glycolysis. But fermentation takes over that pyruvic acid. Rather than continue on this pathway for aerobic respiration, goes on a different pathway. The pyruvic acid, okay, is broken up. Sometimes it forms, depending on the organism, the enzymes, lactic acid. Other times it forms ethanol, okay, alcohol, carbon dioxide. So it just depends on what type of organism. But that's fermentation. Like we said, this is used in many different food products and things that we use. Cheeses are fermented products. Soy sauce is fermented soybean liquid. Yogurts, wine, beer, vinegar. Also household products like nail polish remover, rubbing alcohol. And there's various organisms that are used to produce all of these things. So depending on which of these organisms is going through this process of fermentation will result in different products. All right, but our main concern in this unit is aerobic respiration. Respiration in the presence of oxygen. It requires oxygen. Produces much more ATP than anaerobic respiration. The overall net gain of ATP, it varies a little bit, but it produces 36 to 38 ATP molecules for each glucose that goes in it. So that's far more than were produced in fermentation and anaerobic respiration. And aerobic respiration, okay, it starts off the same way that anaerobic respiration does, with glycolysis, the breakdown of glucose into what? Pyruvic acid, or it gets transferred into pyruvate. You sometimes see it written there. Yeah, that part is glycolysis, glucose to pyruvic acid. Okay? And again, that releases 2 ATP, but that's happening in the cytoplasm. Okay. Then, from there, the pyruvic acid goes through a cycle called the Krebs cycle, or the citric acid cycle, sometimes called. Okay? And this cycle further oxidizes this, this pyruvic acid, extracting more of the energy from it. The final step of aerobic respiration ends with an electron transport chain, like we talked about with photosynthesis, where we take high energy electrons, we allow them to go from higher energy level to lower and lower energy level along the way, using that change in energy to do some work. And finally, it ends with ATP synthase, chemiosmosis, where we manufacture ATP as hydrogen reestablishes its balance and flows down the concentration gradient. So if we look at that, glycolysis produces 2 ATP. Then, as that pyruvic acid goes through the Krebs cycle, okay, we release 2 more ATP. This is also where the carbon dioxide comes from, because we're breaking down these larger carbon molecules and oxidizing them a little bit at a time. It releases some carbon dioxide. Okay, the energy extracted in the Krebs cycle is transferred into the electron transport chain, which produces 34 ATP. So the vast majority of the energy from aerobic respiration comes from this final step. So electron transport chain and then chemiosmosis to produce that ATP. And that gives us our final, that should be carbon dioxide there too, sorry. Our final equation where we started with glucose, we need some oxygen as our final electron acceptor, we produce ATP, we produce some water, and we produce oxygen, or carbon dioxide. So we'll look at these steps in a little bit more detail.
So carbon dioxide is a product?
Yes, of respiration. So this Krebs cycle, you're not really going to be held responsible for the details of the Krebs cycle. Okay? Just like you weren't really responsible for the details of the Kelvin cycle in photosynthesis. But we'll talk about what's actually going on here so that you have some idea about what happens. So one glucose molecule in glycolysis gets broken up into 2 molecules of pyruvic acid. Okay, so this Krebs cycle repeats 2 times for each glucose. Uses pyruvic acid and produces several things. It produces ATP, produces carbon dioxide, and it produces some other molecules, FADH2, NADH. Now, what do you think these molecules do, FADH2 and NADH? Similar to other molecules we talked about in photosynthesis. What was the one we talked about in photosynthesis? NADPH. This is NADH. Very similar, though, and similar functions. What is the purpose of those molecules?
Do they carry and store energy?
Yes, in the form of electrons. They are electron carriers. They shuttle energy and high energy electrons from one part of the cycle to another. That's their purpose. Okay? So if we looked in this cycle, what you have is first you have your pyruvic acid, okay, which is a 3-carbon sugar, or a 3-carbon molecule. Okay. One of those carbons is stripped off. That's one of the ways we get carbon dioxide. Then these 2 carbons are combined with another molecule. Okay? This other molecule is called coenzyme A, and it forms a molecule, and this is a word you should recognize, acetyl-CoA. That's sort of the first intermediate here in this Krebs cycle. So 2 carbons from the pyruvic acid gets combined with a coenzyme A, and we have acetyl-CoA. Okay. These acetyl-CoA molecules get combined. Okay. They go through a couple steps, forming citric acid. 2 carbons are removed, and again we have the release of carbon dioxide. Now we're down here with this. We have this 6-carbon sugar. Get rid of some carbon dioxide. Okay. ATP is produced along the way. We have electrons and hydrogen atoms being transferred to form NADH, FADH2. It requires a couple molecules of water and produces ATP. Okay? And then our final product here is another 4-carbon oxaloacetic acid, which is left to repeat the cycle again. And this is a cycle. And so you need to whatever you start with, you have to regenerate it at the end. Just like when we talked about the Calvin cycle, you have to regenerate those molecules to restart the cycle going again. And you have this repeating cycle over and over again. But the main thing to know here about the Krebs cycle is that you're taking pyruvic acid and you're gradually extracting energy from it. It's being oxidized. We're going to learn about what those terms mean in a second. But it's oxidized, extracting energy a little bit at a time so that that energy can be used to eventually form ATP. Again, just different view of it. And again, pyruvate combined with coenzyme A forms acetyl-CoA. Just goes through this process, okay, releasing carbon dioxide, releasing NADH, okay, eventually going back and forming the end products again, which can repeat this cycle again. Along the way, we form some ATP. Okay. So you see the various intermediates along the way. You don't need to know anything about those. There are various enzymes along the way which allow each step to take place, and so this process repeats. So I've been using these terms, oxidizing. And you'll learn a lot about these next year in chemistry. Okay? We call these ReDox reactions. It stands for oxidation and reduction. Okay? And you could think of it this way. The way often people remember is to remember the word oil rig. You remember that? Oil rig. Oxidation is loss of electrons. Often it means losing hydrogen atoms as well. Okay? And it's removing energy. So when I say glucose is getting oxidized, what that means is each of these steps, okay, it's losing electrons. Okay? Energy is being removed from it. Reduction is a gain in electrons. It's a gain of hydrogen atoms often. It's a gain in energy. So they're opposites. So oxidation and reduction, they always take place together. If one thing is oxidized, okay, that electrons being taken away from it have to go somewhere. And so the other molecule is being reduced. Okay? One compound's always oxidized, the other is reduced. So if you have here an atom that's going to be giving away an electron, okay, it's losing an electron, so it's being what? Oxidized. That electron needs to go somewhere. It doesn't just hang around, it goes somewhere. This thing that accepts it is being reduced. Okay, it's being reduced. It's gaining electron. So think of it, its charge is being reduced. So that's what we mean by oxidized and reduced. Okay, so now the Krebs cycle is taking place. We've extracted glucose and pyruvic acid have been oxidized. We've extracted electrons from them, high energy electrons, in the form of NADH and FADH2. And now we move on to our last steps. The electron transport chain, in this part of aerobic respiration, forms 32 ATP through a whole bunch of redox reactions. And those two molecules are very important, NADH and FADH2. They are the molecules that can carry these electrons from glycolysis and from the Krebs cycle to the electron transport chain. When we talk about these molecules, NAD plus is the oxidized form. NADH is the reduced form. FAD is the oxidized form. FADH2 is the reduced form. So these molecules, NADH, FADH2, those are the ones that are carrying those electrons. Those are the ones that are transporting energy from one part of aerobic respiration to another. So these electron transport chain molecules, they're called cytochromes. They're located in the membranes of the mitochondria. The cristae are those folds, those double membranes that make the mitochondria look like a bean with folds inside of it. Okay? And what happens is as NADH and FADH2 are oxidized, they lose their electrons. They go through this process of gradually from higher energy levels to lower energies. As they do that, they pump hydrogen ions across the membrane, creating this gradient, this proton gradient, where you have a lot of hydrogen ions on one side of the membrane and very little on the other side. So you're building up this gradient. And those hydrogen ions, they want to go back and reestablish their concentration level. We'll take our break in a little bit. Okay? So they establish this proton gradient, and then as those hydrogen ions are pulled through by this enzyme ATP synthase, that is what generates the ATP. ADP is converted to ATP. These hydrogen ions, the final electron acceptor is oxygen. So as oxygen accepts these hydrogen ions and electrons, we end up with water. Again, that's one of the end products of respiration is water, and it comes from these hydrogen ions going through that concentration gradient, electrons being accepted to form water. If we look at sort of just the overview, take glucose, oxidize it, strip off the electrons, send them to some electron carriers, which are NADH and FADH2. Those electron carriers drop off those high energy electrons to the electron transport chain. The electrons gradually lose energy from one step to another, okay? Pumping hydrogen ions to make ATP. Finally, those electrons, as they get down to their lower energy level, they combine with oxygen, they combine with hydrogen, and we end up with water. Again, just another picture to show you this. NADH, FADH2 give their electrons, which go from molecule to molecule through the electron transport chain, all the while pumping hydrogen, more hydrogen. Okay? Okay. Here you see it again, electron transport chain. So here is the membrane, pump these hydrogen ions into the space over here, okay? And then we have ATP synthase, and as those hydrogens come back in to establish that, they turn this ATP synthase enzyme, forming ATP. Okay. And again, it's oxygen that is the final electron acceptor. So that's why oxygen is required for this, because it's going to accept these electrons that have followed that electron transport chain. Just another overview. Start up here in the cytoplasm. Glycolysis takes glucose, transforms it into pyruvate or pyruvic acid. that gets sent into the mitochondria. That pyruvic acid goes through the Krebs cycle. Okay. Krebs cycle produces a little bit of ATP, but most importantly, produces NADH, FADHC, these electron transport molecules. They get sent over to the electron transport chain, pump hydrogen ions out, hydrogen ions come back in, forming ATP along the way. Another view of it. Glycolysis, the first step, glucose to pyruvate, forming ATP. Then we go into the mitochondria, form acetyl-CoA, which goes to the citric acid cycle. Again, produces some ATP. Then, these energy carriers go through electron transport, chemiosmosis to form the rest of the ATP. Those are the various steps. Glucose, glycolysis, Krebs cycle, electron transport chain. The term for this electron transport chain, this producing ATP by the movement of these electrons, is called oxidative phosphorylation. So oxidative meaning we're oxidizing various compounds as these electrons pass. This one molecule loses those electrons. Okay, that's what oxidative. Phosphorylation just means adding that phosphorus onto ADP to produce ATP. All right, so that's a lot of information. But we also have several other things that we'll do. I have an article for you to read, I have a video for you to watch, I have a diagram to give you. So just to reinforce these various parts, because it is fairly complex, but if you break it down into the steps, glycolysis, Krebs cycle, and electron transport, it maybe is not as complicated as it may seem. Okay? So why don't you guys take four minutes or so, and then we'll move on.